Last time we talked about general traits of what is metabolism, how are we breaking down things, anabolism, catabolism, things like that. Today we're going to get into very specific pathways for each, starting first with catabolism. Most microbial metabolism is based around three types of catabolic pathways. So we have aerobic respiration, anaerobic respiration, and fermentation. Usually the nutrient that is being used is going to be glucose. So thinking back a couple chapters, glucose would be that 6-ringed sugar molecule that is a monomer. And usually that's going to be the molecule that's being fed into any of these sorts of pathways. Aerobic respiration is catabolism, so it's breakdown of glucose molecules, or metabolism, in which oxygen is the final acceptor. So it's going to go through a series of different enzymatic reactions to be finally broken down to get ATP out, and the final acceptor for those electrons is oxygen because it's aerobic. So this would be primarily in microbes or just organisms in general that require oxygen. So we are organisms that would do a lot of aerobic respiration. Anaerobic respiration, similar idea, probably still feeding glucose in, still breaking it down with enzymes, getting ATP out, but the final acceptor is going to be a different molecule than oxygen. So it could be a nitrogen-based molecule, sulfur-based, CO3, something along those lines, okay? So this is something you would find in organisms that are anaerobic, do not need oxygen. Fermentation, our third option, is metabolism that is used by aerotolerant anaerobes or facultative anaerobes, basically organisms that can use oxygen, can tolerate it. They don't really care either way. So this pathway will do kind of incomplete fermentation or oxidation of glucose. It's going to be probably one of the more least efficient pathways. Okay, so here's an overview of the three pathways. You'll notice that there are some distinct similarities between them. They all start with glucose up here and feed into the glycolysis pathway. You'll also notice, going back to our last lecture, that there's kind of an integration of different sorts of pathways. So we have linear pathways, and then we have branched pathways, and then we have a cyclical pathway, all kind of integrated together. When I look at this graph, I'm going to show you the key things that I care about. I'm not so much caring that you know every single enzyme in the pathway, but what's the thing going in? What's the name of the pathway? What's coming out? Okay, so things like, oh, aerobic respiration starts with glucose and then it goes to glycolysis and we get NADH out and ATP. And then that is going to feed into the Krebs cycle and so on from there. Another thing that you might want to know from this graph is, what is the electron acceptor? So in aerobic respiration, that's going to be oxygen because it's aerobic, it's in the air. And if it's anaerobic, it's going to be some other electron acceptor that's not oxygen, like SO4, NO3, something along those lines. And fermentation will just use an organic compound. Another key thing to note from this is, what is the maximum yield of ATP? Because again, ATP is the energy currency of the cell. The cell wants to make as much money basically as possible. So we need to know how much we're going to get from this reaction. Aerobic respiration makes 36 to 38 ATP, anaerobic 2 to 36. This one has a lot more variation based on what's being fed in where. Fermentation is much lower. It only nets 2 ATP. So you could see how this is going to be much more costly. You're going to have to be feeding in a lot more glucose to just get a small amount of ATP out. Whereas over here, you feed 1 glucose in, you get a ton of ATP out. So this one's much more efficient than this one. Okay, so we're going to zoom in on glycolysis that each of these three pathways has. As earlier stated, it starts with glucose, which is a 6-carbon molecule. It's then going to get rearranged into a fructose 1,6-disphosphate, and then broken down into 2 3-carbon structures, and then finally into pyruvate. So big takeaways from this graph for me are glucose is what's getting fed in. I'm going to get 2-pyruvate out at the end. Another thing to note is this side part over here. We have to invest 2 ATP at the beginning. So we're kind of in debt at the beginning to get the reaction started, but then we get 4 out at the end. So 4 minus 2, we net 2 ATP and 2 NADH. Pyruvate is critical for aerobic respiration, anaerobic respiration, and fermentation. It's in all three. And the pyruvate molecule here at the bottom is going to be the one that feeds into the next cycle, which is the Krebs cycle. There it can be further modified or it can be fermented. The Krebs cycle is sometimes referred to as an energy wheel. It's a metabolic cycle that transfers energy we go from pyruvate to acetyl-CoA, and then by going around the cycle, we can get NAD plus and FAD out. These should look familiar from earlier in the chapter when we were talking about different shuttle molecules. These are ones that can transfer electrons. When we think back to the last lecture on like oil rig, oxidation, reduction, these are going to be the molecules moving electrons back and forth. The main products or the net that you get out of the Krebs cycle are 2 ATP, 6 NADH, and 2 FADH. So again, we can think through and go, okay, I invested 2 in glycolysis, 2 ATP. I got 4 out, so that's a net of 2. And then I go through the Krebs cycle, I get 2 more ATP out. I would be careful to know exactly how many of these molecules are produced at each major cycle. Okay, so if we look a little bit more, you get pyruvate coming in. Again, it's happening twice because we made 2 from that one glucose molecule. It feeds in and becomes acetyl-CoA, and we go around the cycle here, and you can notice some key molecules that are produced. We have carbon dioxide, ATP, and NADH and FADH are produced as well. Okay, so first step, glycolysis. Second step, the Krebs cycle. Our third major step where we're really going to get a lot of ATP out is the electron transport chain. This is the processing mill for electrons and hydrogen atoms, the spot that we're going to make the most ATP. I guess you're really not making it so much as you're harvesting it because energy cannot be created or destroyed, but we're still collecting it basically. The electron transport chain is the final step for both aerobic and anaerobic respiration, but it is not, you'll notice that fermentation is missing. But aerobic, anaerobic both go through the electron transport chain. It'll just look a little different between the two. The electron transport system consists of a chain of redox carriers. So these are going to be protein molecules in a membrane that are going to be able to receive electrons and pass them along. And they can receive them from NADH, FADH2, these like shuttle molecules that were generated earlier. And then they can pass them from one molecule to the next until we can finally get to the ATP synthase that's going to turn and make a whole bunch of ATP. In aerobic respiration, the step that finalizes the transport process is the acceptance of electrons and hydrogen by oxygen. Water will be produced, oxygen is consumed. So this is in aerobic respiration, we have to have oxygen present. Some of the molecules that will help with the electron transport are going to be molecules like NADH dehydrogenase, flavoproteins, coenzymes, cytochromes. So there's some enzymes. You'll notice this ACE ending that we talked about earlier, some other proteins that are going to be passing the molecules along. ATP synthase, or the enzyme that's located in the mitochondrial cristae, is the one that's going to harness these hydrogen ions and actually synthesize the ATP. Oxidative phosphorylation is the synthesis of ATP using energy that's given off during the electron transport chain. Okay, so if we look in here at the electron transport chain, you'll notice first, this does not look like a mitochondria. And I really want to highlight this because this is a common misconception. Prokaryotes, we talked about earlier, talked about all their cell parts, they don't have a mitochondria. They don't have any membrane-bound organelles. So where are they going to this metabolism, have their electron transport chain? Their electron transport chain is going to be in their cytoplasmic membrane. So we have our cell wall out here with like peptidoglycan and things like that. We have our lipids, and we're just going to have these cytochromes or NADH dehydrogenase or the flavoproteins or whatever's in their electron transport chain is going to be inserted into their cytoplasmic membrane. And it's going to behave a lot like it would in the mitochondria in a eukaryote, where we're transferring electrons from these carrier molecules and they're kind of getting bounced along from all the carriers, and then they're going to go through the ATP synthase and go and add phosphate from the ADP and make it ATP. And then the hydrogens that are left over are going to go onto the electron acceptors, and whether it's aerobic or anaerobic will determine whether it's going to be oxygen or something else. Okay, but again, big takeaway, prokaryotes do not have a mitochondria. Therefore, their electron transport chain is going to be found in their cytoplasmic membrane. Okay, so that said, we do know that not all microbes are prokaryotes. So if you're a microscopic eukaryote, the electron transport system is going to be inside the inner membrane of the mitochondrial cristae. So it's going to be the same as what you learned in Bio 1, where you have the little cristae folds. You have your inner membrane of the mitochondria, the outer membrane, inner membrane spaces, and the little cytochromes and flavoproteins are all going to be stuck inside this inner membrane. It'll work the same way, basically.